In this video, we will discuss about how this FAS signaling can affect this persistence of CAR T cells. Though we have covered these things in when we are discussing our previous paper, but this paper also covered this interactions of the FAS signaling and the persistence of CAR T cells. And here, how they did it, they inject the cancer cells in those mice and then observe this Ki-67 marker, which is the marker for the proliferations of the cells in this mice. And what they are able to see is that so we will start with this panel A. This panel A, we can see that this PMEL Ki-67 expression, since they transduce this PMEL cells, so they can see. So they use two different conditions. First one is the wild type conditions where FAS signaling is not been disrupted, and in another conditions where they remove the intercellular part of the FAS signalings. So when they remove the intercellular part of the FAS signalings, they can see that those cells are able to grow faster compared to the case where FAS signaling are present. And thereafter, so here they do not introduce this tumor cells. We directly introduce the these PMEL cells, which can be identified using this Thai 1.1 reporter. Thereafter, they also checked in the panel B. They checked the relative abundance formations, or comparatively how much which kind of PMEL cells expressed more. So what they are able to see that these cells that have this FAS signalings knockdown, intercellular FAS signalings knockdown, are able to express much higher and to around 50 folds when it reached at its peak, and whereas the PML cells with this FAS signaling pathways not been disrupted are expressed to a very low extent. And when they check these T cells over a period of time, they can see that the T cells which has this Thai 1.1, that is the transduced T cells, goes down with time, and this could be because of the replacement of the T cells with this endogenous T cells which are being produced by the mice. And so this is the main comparisons that the researchers has done. And thereafter they also check here the expression levels of the different expression levels of the different kind of PMEL cells, that is one with FAS that is present in the intercellular domains as a wild type, and in another case where FAS is been intercellular domains has been knocked down. Here we can see that those amount are relatively higher and has this life-1 signalings, whereas the cells that has the empty vectors are life-1 negative. That can be seen here. It is clear. And thereafter they also checked this, you can say that whether this PMEL1 is been expressed in the site where tumors are usually found, that is in the spleen and the tumor sites. And what they are able to see is that the PML cells with FAS signaling wild type has been goes down after 7 days of the transfusion in the spleen, and also in the tumors the amount of PML cells with this FAS signalings present as a wild type in the intercellular domains goes down. Comparatively, when they check it for the PML cells with this intercellular FAS domains has been knocked down, its amount goes up, both in the case of spleen and also in the case of tumors. So this could be because of the non-triggering of the intracellular FAS signalings, thereby these cells doesn't undergo apoptosis, whereas the cells that has the wild type FAS intercellular domains signaling pathways active, those cells goes down because of the apoptosis. So since the amount of these PMEL cells are higher at the site of this tumor infections, there is more possibility that their overall results should be much higher. Their overall results or their overall immune response against the tumor cells will be much higher compared to the case where PMEL cells are present in a low amount. This is how they present the interactions of this FAS signalings and this persistence of CAR T cells, and what could be its impact in the case where these CAR T cells interacts with the tumor. That's all for this video. If you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.